Welcome, folks, to microbiology. In this class, we're going to be looking at the microorganisms that live in and on and around us all the time. They've co-evolved with us and within our ecosystems, generally speaking, to produce symbiotic relationships that allow for the health of us as an individual and also our ecosystems and the world around us. Now, there are times when they also produce pathology, and that's important for us to understand as well. And so in this class, we'll try to get an understanding of who microorganisms are, what are their characteristics, and how do they interact with the world around us. And indeed, they do dominate the world around us. Even though we can't see them, their small size is kind of deceptive because there are so many of them. Just in and on the human body, there are more than 40 trillion microorganisms that make us not an individual, but a community. And so being able to understand them and how they impact the world is really essential. And as we're coming out of a pandemic, we already know that even non-living microbial agents like viruses can restructure global systems, right? And so in our discussion today, we're going to talk about who are microorganisms, what different categories are of microorganisms, and what characteristics do they have. We'll talk a little bit about the history of microbiology, and we'll talk about the scientific method as the main tool for understanding microorganisms. So let's jump into the microbial world for a second, right? If you were to look at pond water, you would see microorganisms, some living and non-living things. You would see debris or crystalline salts or sand, but you will also see microorganisms that were alive. And so you can see some here. Some of them would be complex. Some of them would be more simple. And it would open your eyes to a group of organisms that are constantly around us, okay? And so I'd like to read for you from the book, I Contain Multitudes, which is also assigned reading for this first week, a little bit more about the microorganisms in our world. Okay? All of us have an abundant microscopic menagerie, collectively known as the microbiota or the microbiome. They live on the surfaces, inside of our body, sometimes inside our very cells. The vast majority of them are bacteria, but there are also tiny organisms, including fungi such as yeast and archaea, a mysterious group that we'll meet later. There are viruses too, in unfathomable numbers, a virome that infects all of the other microbes and occasionally host cells too. We can't see any of these minuscule specks with our naked eye, but if our own cells were to mysteriously disappear, they would perhaps be detectable as a ghostly microbial shimmer outlining a now vanished animal core. We are colonies. Ants live in colonies. They can number in their millions. But every single ant is a colony unto itself. When Orson Welles said, "We're born alone, we live alone, we'll die alone," he was mistaken. Even when we're alone, we are never alone. We exist in symbiosis, a wonderful term that refers to the different organisms living together. Some animals are colonized by microbes while they're still unfertilized eggs. Others pick up their first partners at the moment of birth. Then we proceed through our lives in their presence. When we eat, so do they. When we travel, they come along. When we die, they consume us. Every one of us is a zoo in our own right, a colony enclosed within a single body, a multi-species collective, an entire world. The Earth contains a variety of different ecosystems: rainforests, grasslands, coral reefs, deserts, salt marshes, each with its own particular community of species. But a single animal is full of ecosystems too. Skin, mouth, guts, genitals, any organ that connects with the outside world. Each has its own characteristic community of microbes. Indeed, microbes are everywhere. They live in the water of the deepest oceanic trenches and in the rocks below. They persist in the belching hydrothermal vents, boiling springs, and Antarctic ice. They can even be found in the clouds, where they act as seeds for rain and snow. They exist in astronomical numbers. Actually, they far exceed astronomical numbers. There are more bacteria just in your gut than there are stars in our galaxy. In fact, we are legions, each and every one of us. Always a we, never a me. Forget Orson Welles and heed Walt Whitman. I am large. I contain multitudes. And so you can see that really microorganisms are constantly around and within us. And to be able to look at them, we need special tools. And so in our discussion of history of microbiology, you'll learn about von Leeuwenhoek and others that have developed over the years theories and tools that allow us to understand microorganisms. Particularly one of real importance is the microscope. Here's von Leeuwenhoek's microscope, and we now have more complex compound microscopes, even electron microscopes that allow us to see these organisms and learn more about who they are. So let's discuss more who are these organisms, some of the ones that you saw in that video that you were watching, and what are their characteristics generally. And then, of course, throughout the term, we're going to continue to build on that understanding. So first, let's review here the domains of life. So if you remember from previous classes, there are 3 domains of life, broad categories in which organisms kind of are classified, and each domain has a significant amount of diversity, but it does have themes. All of the organisms within it have similar themes, especially in terms of their cellular structure. So we, as humans, exist within the domain Eukarya. The cells of domain Eukarya tend to be complex. They have compartments within them. And because of that complexity within the cells of Eukarya, they can be unicellular, yes, but they can also be multicellular organisms like us. Then we have bacteria. Bacteria, as well as the third domain, Archaea. Both of these are considered prokaryotes, which have very simple cell structures. They don't have complex compartments, and they are always unicellular. Sometimes they form colonies, but never a full-on multicellular organism. Okay? And so bacteria and archaea are significantly smaller than eukarya, but they are the vast majority of the organisms that we find on Earth. And we're going to continue throughout this term really to focus on bacteria and archaea and to come back to some of their characteristics. Okay? So let's look a little bit at the microorganisms that are within these different categories. So eukaryotes do have some microorganisms, both unicellular and multicellular, that are fungi, protozoa, algae, and small animals. And then we said that bacteria and archaea are always unicellular. We're going to come back to those at the end of our discussion today, actually. So let's start with fungi. Fungi are eukaryotic. They have complex compartmentalized cells, And oftentimes they're filamentous, so they actually grow in filaments. They include both molds, which are multicellular but do have some really beautiful microscopic structures, and yeasts, which are unicellular and are important both within the microbiome or the body of the human being, as well as important for food production. Here you can see a mold and some of these beautiful reproductive structures with spores on them. And then these are yeasts, and these yeasts are actually reproducing through budding, which they have a small daughter cell that actually is a clone of the parent that comes off of the parent cell. Another group of eukaryotic microorganisms are protozoa. Protozoa are protists, or from a group of organisms within eukarya called protists, that are animal-like in their features and tend to be single-celled organisms. They're also heterotrophs, which means that they don't produce their own food. They get nutrients and food from their environment and from other organisms. And they can reproduce both sexually and asexually. And so here are some organisms like amoeba and various types of ciliates that are single-cellular organisms that live in pond water, for example. And so if you were to look at the pond water that we have in the lab, you would see many very interesting different types of protozoa, but also algae, which we'll talk about in a minute, and even small animals. Speaking of the lab, here is a video that we actually captured a couple years ago in the lab, where we have an amoeba, which is a single-celled predator that is preying on some other protists or protozoa in the environment. This is specifically called a paramecium. And so you'll see that the amoeba actually has what are called pseudopods, which are arms that reach out to try to engulf some of those other organisms in its environment. And so you can see even the internal structures of the amoeba are moving along as these pseudopods grow to be able to capture some of these organisms. Okay. Can see even at the terminal ends over here, they're actually trying to capture multiple prey at once. And in this area, they do actually capture one of those protozoa. And now it's being engulfed, and you can start to see it even stop moving as then it's being incorporated into the cell of the amoeba and digested. So even though this organism is only one cell large, it does have a cell, but it's only one, then it is able to be kind of very, very active and interactive within its environment. Also within the category of protists, where we have animal-like protists, we also have plant-like protists, also called algae. These tend to be unicellular or multicellular. So for example, kelp is a multicellular algae, and they're photosynthetic. So you can see here, this is Spirogyra. These are different types of diatoms, and these are all photosynthetic organisms, usually living in aquatic systems that are eukaryotes, so they are complex within their cell structure, and they are able to do photosynthesis as well. Okay. There are even some really beautiful bacteria that are photosynthetic, and actually for a long time were confused with algae, but it is clear that they have much simpler cell structures, even though they have a lot of similarities in the way they interact in the environment with some of these algae. And those are specifically called cyanobacteria. We'll talk more about them throughout the class, but specifically in chapter 11 as well. We also have small animals that exist in and on and around us, and these tend to be multicellular heterotrophs. Even though they're microscopic, they are essential to our body, but also environmental systems. And so there are fewer animals that live on our body in and on us, but within the environment, there are many. Some that live in and on us are parasites. But we even have like Demodex, which is a face mite, which literally just lives in the hair follicles on our face. And so this is one of the small animals that is actually a symbiont with us as humans. And you can see it here. It looks kind of similar to a tardigrade. Let me just show this to you as it's active here. It looks similar to a tardigrade in its kind of fore regions, but it has this longer tail. And these tend to live in the follicles on our face naturally in healthy human microbiomes. And then when we sleep, they come out and kind of recycle or eat dead skin cells and things like that, help us clean up, and then they go back into the hair follicles. If there's too many of them, that can cause issues, but it's really normal and natural to have some of these. Okay. And so those are some of the eukaryotes that are microorganisms in and on and around us. I will tell you that in this class, we don't focus on eukaryotes. We tend to focus on prokaryotes in this class. So we'll mention eukaryotes here and there, but really our focus are bacteria, archaea, and then microbial agents that are non-living, which we'll talk about in a minute. Okay. So bacteria and archaea. Bacteria and archaea are two separate domains, and they are diverse, each in their own right. And so we'll talk about them, especially in chapter 11, but throughout the term, we'll talk about them. And they are all unicellular, and they lack complexity within their structures, within their cell structure. But they are different in how their anatomy, their cellular anatomy, and they have different types of organisms and categories within. But if you were just to look at bacteria and archaea, they look very similar to one another, and they are very, very small. So what you're looking at here is actually two human cheek cells. Here's one. Here's the other. Here's the nucleus of the larger human cheek cell. Here's the nucleus of the other human cheek cell. And then you have small bacteria around, okay? And so you can see that just this area of the image, it's 20 micrometers. So these are about 1 micrometer in length or size. They're very, very small, and they're very small compared to this eukaryotic organism. And so they can be difficult to see, and we do need various types of techniques, methods, assays for us to be able to probe really how they're metabolizing, how they're interacting with other organisms in their environment, so on and so forth. And so that is going to be a focus of our laboratory techniques in the class as well. And you can see some of the diverse structures that we can see. And then we continue to add to that with our lab techniques and understand how and probe really how they're interacting with their environment. Okay. So there's lots of diversity in the different types of microorganisms that are in and on and around us. One thing that I want to note, though, is that there's something called the central dogma in biology. And that is, no matter which domain that you're working with, eukarya, archaea, bacteria, we always have a similar genetic kind of blueprint, which is DNA, a double-stranded DNA, that is translated, or actually, a DNA, which is copied to RNA before it's translated into proteins. And so that double-stranded DNA is the blueprint that remains separate and unique from the metabolic functions of the cell. If it's in a eukaryotic cell, that's separated in a nucleus. If it's in prokaryotic cells, that's just separated in kind of a region of the cell on its own. Then there are RNA, single RNA copies that are made of the specific genes that we need or that organism needs. And then those RNA copies are translated into proteins that are functional throughout the cell. And so all of these living systems really rely on that dogma, which allows us to use prokaryotes, for example, like E. coli, to sometimes understand more complex living systems like multicellular eukaryotes. Also, because of those similarities, we can actually monopolize on some of these techniques where we can use recombinant DNA technologies to actually utilize genes from one species in another species for industrial purposes, for example. And so we'll talk about that throughout the course, but just remember that no matter which microorganism that you're working with, in fact, no matter which macro or microorganism that you're working with, we all rely on this same central dogma in the way in which our genetic information is processed and expressed. Now, with that said, we have a couple of microbial agents that are not really considered microorganisms. They're not really considered alive, although there is a little bit of a discussion around that, and we will continue that discussion in this class, specifically viruses and prions. And so viruses and prions, they interact with the central dogma. They interact with living systems. Oftentimes they hijack living systems, but they themselves are not fully functional, fully metabolizing units, okay? And so they can't really exist on their own, but they do have a huge impact on living systems, as we already know coming out of this pandemic, okay? So these are actually viruses that infect bacteria, and so you can see a virus, or what's called a bacteriophage, a phage or virus of a bacterium, that's actually, it's injecting its genetic material into this poor bacterium that the virus will then take over its machinery, the bacterial machinery, and regenerate itself via that machinery. Okay. And so, as we're looking throughout this term at these different types of microorganisms and microbial agents, like we said, we're going to focus on prokaryotes, bacteria and archaea. We're also going to focus on viruses and prions, not so much on eukarya, although we will have some discussions here and there about them, and you will be able to utilize them for your term project as well. Okay? So in order to understand these very small systems, we need a significant amount of tools, and we need a really good understanding of what's going on at a micro level. And so the next part of this chapter is a review of the history of microbiology, a history in which microbiologists and scientists, and just folks generally, are trying to understand these microorganisms and microbial agents that at one point were almost impossible to see. And then we developed tools that allow us to see them with the microscope. We've also developed tools to allow them to see how they're metabolizing or what their work is within these microbial settings. And so questions like, where is life coming from? Is it spontaneously generated? What is going on when food spoils? What is the cause of disease? So on and so forth. And so what I'd like you to do is, instead of us going through the slides together in this lecture on the history of microbiology, there's just this really well done crash course that covers the history of microbiology. That's linked in the description below. Go ahead and jump into that link and review the history of microbiology there. Another thing that I want you to do as you're going through that history of microbiology is think about two things. One, what are the microorganisms that you interact with in your normal daily life? And two, what are some of the historical figures, historical theories, or series of understandings and events that come out in that crash course that are really interesting to you, maybe to your career, or maybe to something that you've experienced in your life or your world around microorganisms. And those 2 points are going to be the basis of our discussion for this chapter that you'll publish to D2L. So you'll talk about how you've interacted with various types of microorganisms, some of which we've mentioned here, in your life or in your career. And then you'll talk about some of these key historical concepts that really stood out to you. You can choose 1 or 2 as you watch the crash course video. Okay, so go ahead and open yourself a note page so that you can start your discussion post, and open the crash course that's linked below and review the history of microbiology. And then you can come back into the lecture and we'll continue our discussion here. Welcome back. So I hope you were able to engage with that video and learn about the history of microbiology and start to think about that history and how it relates to microorganisms in your daily life or in your career. Okay. So I want to remind us that the kind of the last point for this lecture is that I want to remind us that all of these different examples of microbiologists in history remind us of the procedure or the steps that we take to understand scientific questions. In microbiology and physics and chemistry, whatever it is, some of these questions that you see in front of you that were engaged in that video that you watched. And so this is called the scientific method. And I'm sure you've interacted with scientific method in prerequisite classes to this course, but I do want to review together the scientific method here. Okay. So within the scientific method, we start with an observation in the natural world. You saw many examples of that, observations related to food spoilage or disease around a water source, so on and so forth, or aseptic technique in that video of the crash course video. And then those scientists asked specific questions and started to develop, based on their understanding and the understanding of other scientists around them, a hypothesis. And so that hypothesis is a clear statement of what might be expected when 2 variables are in interaction with one another. Okay, so if this water source is contaminated, we would see more folks getting sick around the water source compared to another water source, or compared to shutting off that water source, right? And then you're able to take those 2 very clear variables and set up some sort of way to test that experiment or that hypothesis. You might be able to test a hypothesis using a controlled experiment where all the variables or almost all the variables are controlled and participants are randomized. You might not be able to, though. It might be unethical, although historically within science, there have been issues with the way that we answer questions in terms of ethics. We will continue to revisit that throughout the term as well. It might be unethical, or it might be a question that's just not easy to produce an experiment around. For example, a really rare disease where you only have a couple of case studies to work with, right? However you're able to do it, optimally a controlled experiment, maybe a case study, or maybe just an observational study, then you can go through and try to ascertain the relationship between those 2 variables. If you're able to do a controlled experiment, then your conclusions are going to give you an idea of specifically a causal relationship between those 2 variables. If you're not able to do a controlled experiment, then you're just going to learn about the correlation between those 2 variables, and those correlations might be impacted by other variables around or within that system. Once data is collected, it has to be interpreted, analyzed, and presented, okay? And so this is in scientific papers that have specific standardized structures so that different scientists around the world can learn about the question, the background information, the methods that were used, the results that were the results of the experiment, and then discussions of those results. And it is really essential that scientists share and publish their results, especially in easily accessible ways, because it is in that way that those results will then be meaningful to other scientific research or general application. And so in this class, we are going to practice not only doing scientific research, but various ways for us to present our scientific research in scientific format and writing, but also in artistic and diagram and other formats as well. Okay? And so that research, when it's published and then is accessible to other scientists or other folks around you, then it can start to inform other work related to that same phenomenon. Okay? Now, there's another really important distinction that you might already know, but we want to just remind, is that once you have many different hypotheses that are revolving around a similar phenomenon that tend to support various aspects of a pattern within that phenomenon, then we can start to develop what are called theories and laws. And you saw some of those in the video, like the cell theory and the germ theory, theory of evolution. All of these are theories, are concepts, are ideas, foundational core ideas that allow us to understand systems generally, okay? So when we use the word theory, just in general lingo, it's much less, it's more kind of like hypothesis, a thought. But when we're using theory in a scientific realm, we're talking about an idea that is really a foundational and core understanding that is near fact, okay? Hypotheses, theories can change, but generally speaking, we're talking about a foundational core idea that is near fact and nearly agreed upon, okay? And so that's really essential when we're starting off a scientific course to understand and get into that framework, okay? So that's essentially the core parts of chapter 1. I want to remind you to go ahead and complete your discussion post and actually post it to D2L, as those are going to be important for you to accumulate points over the term. So that's chapter 1 for us. There is some detail in chapter 1 in the book and in the second chapter of the I Contain Multitudes reading that give you some more detail about what we discussed and talked about in lecture today. So I definitely direct you to those readings to digest them, to get some more information and detail from them. So what we've covered in class today is an introduction to the beautiful world of microorganisms and some of the microorganisms that exist within our world. We've talked about the history of microbiology and some of the key scientists and theories and explorations that have brought us to this point in microbiology. And then we just took a minute to remind ourselves about the scientific method and how it works, because that's really going to guide our understanding of the various concepts in microbiology. And it's also going to guide our labs that we do together in person. So thank you for listening. I'm so excited to have you in this class, and I look forward to meeting you both in person in the lab and continuing our lecture series together here on our channel. So thanks for listening. Take care. See you soon.